Welcome. Welcome to this session of steroids. Today in this session we are going to discuss about the nomenclature, stereochemistry, metabolism of these steroids. What are these steroids? Actually, these are the part of the endocrine systems. They are the secondary metabolite of the endocrine system. The endocrine system, which is a chemical messenger system that comprises of the hormones which are released from the internal glands of an organism. After releasing from the internal gland, they comes directly into the circulatory system, and finally they regulating the distinct target organs. In humans, the major endocrine glands are the thyroid gland and the adrenal glands. These are very specialized endocrine glands, and many other organs that are the part of the body system, they have secondary endocrine function also, like bone, kidney, liver, heart and gonads. Just like, for example, the kidney secretes the endocrine hormone erythropoietin. These hormones, they can be amino acid complexes, they can be steroids, eicosanoids, leukotrienes or prostaglandins. Today in this session, we are going to talk about these steroids. Steroids. These steroids, they are the lipophilic low molecular weight compounds which are derived from cholesterol that play a number of important physiological roles in the body. The steroids hormone, they are synthesized mainly by the endocrine gland such as gonads, which are testes and ovaries. Also the adrenal gland, and during the gestation by the fetoplacental unit. And where they are secreting? They are secreting into the blood circulation, or they are releasing into the blood circulation. These steroids, they act both on the peripheral target tissues and the central nervous system. A very important function of these steroids hormone is to coordinate the physiological and behavioral responses for specific biological purpose, just like reproduction. If you see the structure of these steroids, they are biologically active organic compounds with 4 rings arranged in a specific molecular configuration. Actually, these steroids, they have 2 principal biological functions as we discussed. First is it is an important component of the cell membrane which alter the membrane fluidity, and second is as a signaling molecule. There are hundreds of steroids which are found in the plants, animals and even fungi, insects also. These all steroids, they are manufactured in the cell from the sterol, just like sterols, lanosterols, cycloartinol. So these lanosterol and the cycloartinol, they are derived from the cyclization of the triterpene squalene. Now we can see in the structure, in the given structure of the steroid, there is a core structure which is typically composed of the 17 carbon atoms which are bonded in 4 fused rings. See the structure, there are 3 6-member cyclohexane rings, A, B and C, and 1 5-member cyclopentane ring, which is the D ring. Steroids vary by the functional group attached to this 4 ring and by the oxidation state of the rings. How they differ from each other? By the functional group which are attached on the D ring, that is the 4th ring, and the oxidation state of the other rings also. Sterols, they are the form of the steroids with a hydroxy group at position 3 and a skeleton derived from the cholesterol. At position 3 there is an hydroxy group, so this is known as the sterol. Moving to the next part is the classification of steroids. This classification is based on the function and the structure of the steroids. Based on the function of the steroids, these steroid hormones, they are classified into corticosteroids. They are also classified as sex steroids. In the corticosteroids, they are glucocorticoids like cortisol, which is a corticoid whose function is include the immunosuppression. Some are mineral corticoids like aldosterone, which is a mineral corticoid and help regulating the blood pressure through water in electrolyte balance. The second is the sex steroid, which are progestogens. These progesterones, they like the progesterone, which regulate the cyclical changes in the endometrium of the uterus and maintain a pregnancy condition. Androgens like testosterone, which contribute to the development and maintenance of the male secondary sex characteristics. Also, estrogen is a part of sex steroids, which is contribute to the development of maintenance of female secondary sex characteristics. Now, if we see by the structure, you can see this table. This table is a class and some example with number of the carbon atom. If the carbon atom, number of total carbon atom is 27, so they are considered the cholesteins, which are example is cholesterol. With 24 carbons, they are choline, example the cholic acid. With 21 number of carbons, progesterone like pregnine, and 19 carbon, testosterone comes under the androgens. With 18 number of carbon, estrogens, example is estradiol. This is one of the very important classification based on the structure as well as on the function. Now moving to the next slide, nomenclature of the steroids. You can see that this is the cholesterol, which is a sterol. Now the guanine, which is a cyclopentanoperhydrophenanthine ring moiety, which is comprises of these 4 ring moiety A, B, C and D. Overall, this is a cyclopentanoperhydrophenanthine moiety, and this is the simplest steroid and the nucleus of all steroid and the sterols. With this structure, we can say that these steroids, they are the compound which possess the skeleton of cyclopentylphenanthine, or a skeleton which is derived from or by one or more bond scissions or ring expansion or the contraction. Now, in case the methyl group are normally present at carbon 10, you can see that there are number 1, 2, 3, 4, 5, 6, 7, 8, 9 and 10, and then 11, 12, 13, 14, 15, 16, 17. This 17th position is very important. We will discuss it later. So this is basically an alkyl side chain which is present at this carbon 17, and methyl group are normally present at carbon 10 and even also at carbon 13. Steroids, as discussed, these are the steroid carrying a hydroxyl group at carbon 3 and the most of the skeleton of the cholesterols. Additional carbon atoms may present in the side chains at the position of 24, 27, etc. Now, numbering and ring letters. Very important. The steroids, they are numbered and ring letter as in the formula A ring, B ring, C ring and D ring. If one of the two methyl group attached to carbon 25 is substituted, it is assigned the lower number. What does this mean? That at position 26 and 27, if one of the methyl group which is attached to, that is 26, for example 26, is attached to the carbon 25, if it is substituted, then this 26 number will get the lower number. If, for example, the 27 number gets substituted, then this 27 becomes the 26 and the above one becomes the 27. And if both are substituted by, for example, one substituted by the ethyl and second is substituted by propyl, then it will carry the alphabetically order, which are an alphabetically order assign the lower number. That means ethyl will get 26 and propyl will get 27. It's very important to mention over here that about the absolute configuration of this moiety. The absolute stereochemistry of steroid actually is defined by the parent name of some chiral centers, just like by the alpha, beta, R or S or some other centers. So when the configuration at one or more center is not known, if the configuration is not known, then this is indicated by a Greek letter which is known as xi. Now about the orientation of this molecule. Just like you can see that these are denoted on a paper. Now if we see the paper, then it is normally oriented as like this. Now an atom or group which is attached to a ring, just like it is shown in the structure, that the orientation, if it is above the plane, it is known as the alpha, and if it is below the plane, it is known as the beta. Oh, sorry. The bond which are above the plane, they are known as the beta, and the bond which are below the plane, they are known as the alpha. As you can see in the structure that there are broken lines. These broken lines, they are the alpha. It means they are below the plane. They are below the plane, and they are hedges which are these solid lines, right? Thickened lines, they are above the plane. They are known as the beta. Above the plane beta, below the plane alpha. And there are some wavy bonding, and that this bonding show that the configuration is not known. So this wavy lines, they are indicated where the configuration is not known, and when the configuration is above the plane, they are the beta, and when they are below the plane, they are known as alpha. If you see the structure, and so they are basically the carbocycles. So now with the simplest steroid is the guanine, and in which the parent tetracyclic hydrocarbon without the methyl group at carbon 10. You can see the structure over here. In this structure, the parent tetracyclic hydrocarbon is without the methyl group at the carbon 10 and carbon 13 position. There are two hydrogens, right? One at carbon 10 and one is carbon 13. And without a side chain at carbon 17. So this structure is known as or named as guanine. And when the hydrogen is above the plane and below the plane, the configuration is 5-alpha guanine and 5-beta guanine. The first is the 5-alpha guanine, the second is 5-beta guanine. Estrane. Estrane, in this the hydrocarbon with the methyl group at carbon 13, but without a methyl group at carbon 10 and without a side chain at carbon 17. Right? So hydrocarbon with a methyl group at carbon 13 but without a methyl group at carbon 10. So at carbon 10 there is no methyl group, and at carbon 13 there is a methyl group, and without a side chain at carbon 17, and this is known as or named as estrane. First is the 5-alpha estrane based on the configuration of the bond, and second is the 5-beta estrane. The next is androstenedione. In this, the hydrocarbon with the methyl group at carbon 10 and carbon 13, at both positions, carbon 10 and carbon 13, but without a side chain at carbon 17, it is known or named as androstenedione. And the first is the 5-alpha androstenedione, and the second is 5-beta androstenedione. Moving to the next part of this nomenclature, that is the hydrocarbon with a side chain at carbon 17. This is also very important. From this table, it is easily identified that at carbon 17, if the side chain at the carbon 17 is CH2, CH3, then it is known as pregnane, and two configurations are 5-alpha pregnane and 5-beta pregnane. The second example is of the choline, 5-alpha choline and 5-beta choline. The third is 5-alpha cholestyne and 5-beta cholestyne, and so on. Now moving to the next part, it is again the how the ring, when there is a changes in the ring, what are the names or prefix we use. So during the structural modification in the steroid ring, when there is a modification in the structure, they are expressed by certain prefixes. For example, number one is the ring contraction by loss of unsubstituted methylene group. If the ring is get constructed by the loss of the unsubstituted methylene group, it is indicated by the prefix nor. And if two methylene groups, then it is di-nor is used. For example, the case is below. The structure is given that 4-nor 5-a androstenedione. The first structure is 4-nor 5-a androstenedione. When there is a ring expansion by inclusion of one methyl group, as you can see in the A group, in the first example, which is 5-nor 5-a androstenedione, the A ring it get contracted, Now in the second structure, see the second structure, the A ring, it gets expanded by one methyl group, right? So then it is prefixes as homo, and if there are two methyl groups, they are known as dihomo. So the example is this 4A homo 7 nor 5 alpha androsten. Why we are saying it the 4A homo because on the A ring it get one ring expansion is by one carbon, right? Why we are saying 7 nor because in the B ring one methyl group is contracted or loosed, that means ring get contracted, so that's why we are using 7 nor 5 alpha androsten. See the example third structure. If the terminal ring is removed with addition of a hydrogen atom at each junction atom with the adjacent ring, it is indicated by a prefix that is des. Now, so we will say the example is des A endocrine. Why we are using this A is because we are talking about A ring, because the removal of A ring, just like in the previous example, the first structure, see the first structure, it is 4 nor 5A androsten because the changes is taking place at A ring, that's why we are saying 5A androsten. That's why in this third option we are saying des A androsten. That means A ring has been removed with the addition of hydrogen atom at each junction. So now in the fourth structure there is a fission of a ring with addition of a hydrogen atom at each terminal group, and that's why it is indicated by a prefix CCO. So this structure, the fourth one structure is 2,3-CCO 5A cholesterol. The previous three were one androsten, and this is cholesterol. Now moving further, the stereochemistry of this steroids. Stereochemistry of steroids reveals them to be a non-flat molecules and hexagonal carbon ring from A to carbon to ring C, and usually assumes a chair form rather than a boat conformation. So they look like a chair rather than a boat conformation. These rings, they are mostly fused in a trans conformation, and the functional groups, they are oriented either in the equatorial or the axial position, as we discussed earlier, the alpha and beta. So the subsequent group above the plane of the molecule, they are said to be beta position, whereas those present below the plane, they are the alpha position. So this stereochemistry is based on the ring junction and the side chain, stereochemistry of the substituent in the side chain, and the stereochemistry of the double bond. It's a very broad topic, so might be we will cover it in the coming classes. But stereochemistry of this steroids is very complicated, but we have to learn some basics, and these are which we have discussed now. They are some of the basics of the stereochemistry of the steroids. Moving to the next part is these steroids, they are usually identified by the trivial names. Example, cortisol, Why is it so? Because it is sometimes very confucius. That means they lead to confusion in some cases during the naming of these steroids. So that's why rather than taking the trivial names, it is recommended that all we know these steroids by the trivial names, right? But it is recommended to use the IUPAC names. Just like see this example, it is the structure of the testosterone, and testosterone is a trivial name, and the IUPAC name is 17 beta hydroxyandrost 4-ene-3-ol. 17 beta hydroxyandrost 4-ene-3-ol. Now talk about the biosynthesis of steroids. So as a general outline, the adrenal gland produces both androgens and corticosteroids, which are the mineral and glucocorticoids, including mineral and glucocorticoids. Ovaries, it depends on the stage of the ovarian cycle, they secrete estrogens, progestin, and testes, it mainly secrete or produce the androgens. The biochemical pathway involved very similar in all the tissues, but the difference in the secretory capacity because of presence and absence of some specific enzymes. Although the biochemical pathway, they are all similar. Now, if you see the general outline of the biosynthetic pathway of the steroids, which are applicable to all steroids which are secreting from glands, so they are produced from the acetate to the cholesterol, and from cholesterol they further produce some other steroids. They also produce from the cholesterol to progestins, androgens and estrogens. And the last is from progesterone and 17 alpha hydroprogesterone to gluco and mineralocorticoids. There are some enzymes which are involved in the steroid biosynthesis, just like desmolases or lyases, hydroxylases, hydroxysteroid dehydrogenases, and aromatase enzymes. They have different functions. Now, there are some disorders which happens because of the problem in the biosynthesis. A number of endocrine disorders, they occurs because of some specific enzyme defect, like the inability to secrete the normal level of adrenal steroids. They result in congenital adrenal hyperplasia, which further cause the hyperstimulation of the ACTH, that is adrenocorticotropic hormone. In majority of these cases, in this congenital adrenal hyperplasia, this syndrome is due to the 21 hydroxylase deficiency, which is associated with increased adrenal androgenous secretion and partial virilization in girls. Some of the less common adrenal enzyme deficiency involves either 17 hydroxylase or 18 hydroxylase. The other example is defect in the testicular androgen synthesis. This defect in the testicular androgen synthesis because of the desmolase enzyme or hydroxysteroid dehydrogenase deficiency that lead to the male pseudohermaphroditism. This pseudohermaphroditism is a male problem which is due to this desmolase enzyme defect and 17 beta hydroxysteroid dehydrogenase deficiency. These steroid hormones, they are assumed that steroids, they are released into the body, into the blood circulation as soon as they are formed. That is, there are no active transport system or release mechanism. So once they formed or produced, they just come directly into the blood circulation. Secretion rate are therefore directly related to the biosynthetic activity of the gland and to the overall blood flow rate. So there are some steroid, once it comes into the blood circulation, the steroid start to bind with the proteins. Because of their lipophilic properties, free steroids molecules are only sparingly soluble in water. Free steroid molecules. In biological fluids, they are usually found in conjugated form and unconjugated form. In a conjugated form, they try to link to a hydrophilic moiety where the sulfate or glucuronide derivatives, or they bound to the proteins through non-covalent or reversible binding. While in plasma, unconjugated steroids, they are found mostly bound to the carrier proteins, just like binding to the plasma albumin, and they count around 20 to 50% of the carrier protein of the bound to the fraction. Whereas binding to either corticosteroid binding globulin or sex hormone binding globulin, but only very small portion of this steroids which are free fraction is only 1 to 10%. Now the next is in the metabolism of the steroid, that how they are moving into the bloodstream and get metabolized, because this is the peripheral metabolism of these circulating steroids. So because the steroids, they are lipophilic, they diffuse very easily through the cell membrane, and therefore they have a very large distribution volume. With their target tissues, these steroids, they are concentrated by an uptake mechanism which relies on their binding to the intercellular protein, or we call receptors. High concentration of these steroids, they are also found in adipose tissues. Although these adipose tissue are not the target of these steroids. In human, especially man, the adipose tissues contain aromatase activity and seems to be the main source of androgen derived estrogen which are found in the circulation. Remember that most of the peripheral metabolism occurs in the liver and to some extent in the kidney. This kidney, which are the major site of hormone inactivation, because when it's produced, get activated, bind to the target or the protein or receptor, then it will get inactivated also and then eliminated, or catabolism also occurs. The biological activity of a steroid molecule, it depends on its ability to interact with a specific binding site on the corresponding receptor. But in most cases, this biological activity can be directly correlated with the binding affinity. The affinity which is characterized by the binding constant Kd, and also which is the molar concentration required to saturate the half of the available binding site. This is how the binding affinity is measured. It's very important to mention over here that certain class of hormones and particular target tissues, steroids must be converted into active forms. That means the metabolic activation is required, or it is a prerequisite for achieving a range of effect which involve the interaction with more than one type of the receptor. This activation, for example, the first is the conversion of the testosterone to 5-alpha DHT, which is required for its action on the prostate growth and function. Whereas the aromatization, if take place to, and it gives the estradiol 17 beta in the brain, it is mandatory for some of its developmental and neuroendocrine and behavioral effect. While on the opposite side, if we see the parent compound progesterone and its metabolite is 5-alpha DHP. This 5-alpha DHP has no effect on the uterus. While the conversion of the testosterone take place to the 5-alpha DHT, which is the having the action of the prostate growth and function. And this progesterone metabolite, that is 5-alpha DHP, has no effect on the uterus, where it is located. That is where it is located. Now, next is the inactivation and the catabolism of these steroids. The inactivation refers to the metabolic conversion of a biological active compound into an inactive one. This inactivation can occur at various stages of the hormone action, just like the peripheral inactivation, which is done by the liver enzyme. The main site of this peripheral steroid inactivation and catabolism is the liver, but some catabolic activity also occurs in the kidney. Depending on the structure of the starting steroid, there are some following reactions which happens. The first is the reduction of the double bond at carbon 4 and the reduction of an oxo, that is keto group at carbon 3 to a secondary alcoholic group. The second is reduction of an oxo group at carbon 20 to a secondary alcoholic group. Third, oxidation of alpha beta hydroxyl group. Also, conjugation as sulfate with the sulfate of the glucuronide derivatives. These are some of the reactions which occurs during the inactivation and the catabolism of the steroids, which happens in liver as well as the kidney. This is all for a very brief, this was all about the brief introduction about the steroids. In the next session, we will do the drugs which are used in the endocrine system. Thank you.